Welcome to the deep dive. Today, we're going to be tackling the sterile insect technique, or SIT for short. We've got a whole stack of academic papers and book chapters all about using bugs to fight bugs. And you know me, I love this kind of stuff.
It is remarkable how SIT has kind of flown under the radar for so long, quietly revolutionizing pest control, and yet many people have never even heard of it.
Exactly. So our mission today is to unravel the mysteries of this ingenious method for ya. We'll uncover what SIT is, how it works, the history behind it, its incredible successes and the hurdles it faces, and what the future might hold for this very promising way to manage pests. By the end of this deep dive, you'll be a walking encyclopedia of SIT knowledge.
That's the goal. Let's equip you with all the fascinating details.
Okay, let's get started. I've got to admit, the eradication of the New World screwworm fly from North America always blows my mind. It's a truly amazing success story in the world of pest control. And, spoiler alert, SIT was the hero of the day.
The screwworm eradication program is indeed a classic example of SIT's potential. But before we dive into that, let's lay the groundwork. What exactly is the sterile insect technique?
Back to basics then. What have we got?
At its heart, SIT is a pest control method that uses sterilized insects as a weapon against their own kind. It's about releasing a flood of these sterile insects into the wild, where they'll mate with wild insects. But here's the catch. Their offspring will be infertile, causing the pest population to dwindle.
So we're essentially pitting insects against themselves, disrupting their ability to reproduce. It's kind of brilliant, isn't it? But I have to ask, is the ultimate goal always complete eradication? Or are there other scenarios where SIT comes into play?
That's a great question. While SIT can be used to wipe out an entire pest population, it's not a one-size-fits-all approach. It can also be strategically used to suppress pest numbers, contain them to specific areas, or even prevent them from establishing themselves in the first place. This flexibility is what makes SIT so powerful. In fact, Hendricks and his colleagues outlined these various strategic options for using sterile insects in their 2021 chapter on area-wide integrated pest management.
That's fascinating. And now for my favorite part, the history. Who first dreamed up this idea of using sterile insects? It seems almost too clever to be true.
We can thank Edward Nippling for that, a brilliant entomologist. Back in the 1930s, he was studying the screwworm, which, as you mentioned, was a major problem back then.
Now, for those of you who may not be familiar with the screwworm, just how bad was this pest? Paint us a picture.
Imagine a livestock owner's worst nightmare. The screwworm fly was a terror for livestock. Its larvae would infest any open wound on an animal, causing excruciating pain and often leading to a gruesome death. It was a major economic burden and caused immense suffering for the animals.
That sounds horrific. That must have been incredibly challenging for ranchers and farmers dealing with that.
Absolutely, it was a crisis. But Nippling had this truly revolutionary idea. What if we could disrupt the screwworm's reproduction by releasing sterile males into the population?
And that was the spark that ignited SIT.
Exactly. His initial focus was on the screwworm, but the principles he uncovered paved the way for what we now know as the sterile insect technique. It wasn't an overnight success, but Nippling's vision eventually came to fruition.
And then came the remarkable victory in the 1950s, the eradication of the screwworm from North and Central America. That must have been a turning point in pest control. It's incredible to think that a seemingly simple concept like releasing sterile insects could have such a profound impact.
It was a groundbreaking achievement, proving that SIT could be successful on a massive scale. And the screwworm eradication wasn't an isolated triumph. SIT has been instrumental in eradicating other formidable pests as well. Take the melonfly, for example. It was successfully eradicated from the island of Rota in the Mariana Islands back in 1963. And then during the 1980s and 1990s, Japan employed SIT to eliminate the melonfly from Okinawa and its surrounding southwestern islands. These were huge wins for SIT, showcasing its global reach and undeniable effectiveness.
Okay, I'm hooked. But I'm also itching to understand how SIT actually works on a practical level. Walk us through the nuts and bolts, if you will.
Let's peel back the layers and explore the mechanics. It all begins with a deep dive into the target pest's world. We need to become experts in its biology, understand its mating behaviors, how its population dynamics work, and any other quirks or characteristics that make it tick.
So we need to know our enemy inside and out, basically.
Precisely. Once we've mastered that knowledge, we can strategize about how to sterilize the insects.
All right, spill the beans. How do we sterilize these insects? Are we talking about some kind of intricate microsurgery?
It's not quite as dramatic as that. The most common method involves radiation. Exposing insects to a calculated dose of radiation disrupts their reproductive cells, rendering them sterile.
So it's like a super focused tiny scale X-ray.
That's a great analogy. We also have chemosterilants, which are chemicals that can induce sterility. And we're even venturing into the realm of genetic manipulation techniques.
So there's more than one way to disrupt those reproductive processes. But I'm guessing we need a lot of these sterile insects, or it can't just be a handful.
You're absolutely right. Mass rearing is another critical piece of the puzzle. We need facilities capable of producing millions, and sometimes even billions of these sterile insects for release.
Billions. That's mind-boggling. Where on earth do you even begin to house that many insects?
Designing and running a successful mass rearing facility is a complex dance, I assure you. It involves creating a suitable artificial diet that keeps the insects happy and healthy, maintaining the perfect environmental conditions for them to thrive, managing their health and well-being, and of course, ensuring that we're producing top-notch sterile insects.
Speaking of top-notch insects, in their chapter on mass rearing for SIT, Parker and his colleagues really emphasized how intricate and expensive this whole mass rearing process can be. It's not as simple as just churning out tons of bugs, is it?
You hit the nail on the head. The quality of those sterile insects is paramount. If they aren't fit and competitive in the field, the entire program can fall apart.
What exactly do you mean by competitive? What makes a sterile insect a strong contender out in the wild?
Think of it this way. Those sterile males need to be able to outcompete their wild counterparts for mates. They need to be robust, healthy, and irresistible to the females. Think of it as a bug beauty contest with survival of the fittest at stake.
So it's not just about sterilizing them. It's about ensuring they're in peak physical condition too. I imagine there are rigorous quality control checks in place to make sure these insects are up to the task.
Absolutely. We monitor and evaluate their quality throughout the entire rearing process. We're looking at their physical fitness, their mating prowess, and how well they perform under real-world conditions. If those sterile insects are weak or don't measure up in terms of attractiveness, they won't be able to effectively suppress the wild population.
So there's a delicate balance to strike between producing enough sterile insects and maintaining high quality. Now, I'm curious about the flip side of this coin. What are some of the challenges and limitations of SIT? Is it a foolproof solution, or are there downsides we need to consider?
That's a really important question. While SIT has the potential to be incredibly effective, it's not a magic bullet, and there are definitely some hurdles to overcome.
Right, let's unpack those challenges then. What are some of the factors that could make SIT tricky to implement or hinder its effectiveness?
One of the biggest challenges lies in the very nature of the pests we're targeting, their biology and ecology. Some insect species are simply more difficult to control with SIT than others.
So what makes some insects tougher nuts to crack?
Take, for instance, insects that are expert travelers. If they have high dispersal ability, meaning they can fly or move long distances, it becomes much harder to ensure that enough sterile insects are present throughout the entire target area. It's like trying to keep up with a swarm of bees that are constantly on the move.
I can imagine that would be a logistical nightmare, trying to blanket a vast area with enough sterile insects to make a dent in the population.
Exactly. And then there are insects that have developed intricate mating rituals or rely on specific pheromone signals to attract mates. Replicating these complex behaviors with sterile insects can be a real challenge. If the sterile insects can't speak the right love language, so to speak, they're going to have a tough time competing for mates.
So it's like they need to blend in with the in-crowd and follow the right social cues to be successful.
Precisely. And we can't forget about those insects that have cryptic life stages, parts of their life cycle where they're hidden away, making them much harder to target.
Could you give an example of that? What do you mean by cryptic life stages?
Imagine insects that burrow underground or spend their larval stage tucked away inside a plant. These stages are like their invisibility cloaks, making them much harder to reach with SIT. It's like trying to find a needle in a haystack.
That makes sense. It's a lot harder to control something you can't easily see or access.
Exactly. Environmental factors can also throw a wrench into the works. Climate can influence insect development and survival. And if a habitat is fragmented, it can be tricky to achieve uniform coverage with sterile insect releases.
And I imagine public perception and acceptance play a significant role as well. Releasing millions of insects, even if they're sterile, could make some people uneasy, right?
Public perception is a crucial factor. People may have concerns about unintended consequences, especially if they're worried about potential non-target effects on other species or the environment. That's why meticulous planning and responsible implementation are absolutely essential. We need to minimize any risks and be transparent about the process.
It's about ensuring that we're not solving one problem only to create another. And speaking of challenges, What about the financial side of things? I imagine these SIT programs require a substantial investment.
You're absolutely right. Financial and political constraints can be major obstacles. Operating those mass rearing facilities isn't cheap, and large-scale SIT programs need sustained funding and unwavering support to be successful.
So it sounds like SIT is a powerful tool with incredible potential. But it's not a simple fix. It requires careful planning,
Yeah.
scientific expertise, and a long-term commitment to ensure its effectiveness.
Absolutely. But despite these challenges, SIT holds immense promise for the future of pest management. There are some incredibly exciting developments on the horizon.
Now that's the perfect segue, because I was just about to ask about the future of SIT. Are we going to see it used for new types of pests, or are we sticking with the tried and true?
Researchers are constantly pushing the boundaries of SIT, working to refine its application and broaden its scope. One area that has everyone buzzing with excitement is the application of genetic technologies. Tools like CRISPR, a gene editing marvel, and techniques like RNA interference, or RNAi, are opening up a whole new world of possibilities.
So we're talking about cutting-edge science, the kind of stuff that sounds like it's straight out of a sci-fi movie.
It really is remarkable. These advancements could help us develop even more effective and efficient SIT strategies. For example, imagine creating sterile insects that are even more competitive in the wild, or using gene editing to disrupt specific pest behaviors.
So this is like taking SIT to the next level, a souped-up version.
You could say that. Another exciting avenue is the integration of SIT with other pest control methods, combining SIT with traditional approaches, like using insecticides, introducing biological control agents, or implementing cultural practices, can create a multi-pronged attack that's even more effective.
So it's about using the best tool for the job. And sometimes that means combining different approaches for a more powerful punch.
Exactly. And there's a growing movement to expand the use of SIT to tackle new targets, including those pesky disease vectors, like mosquitoes. Imagine the impact we could have on global health if we could effectively control diseases like malaria or dengue fever using SIT.
Now that would be a game changer. I can see how controlling disease-carrying insects could have a monumental impact on people's lives around the world.
The potential is truly extraordinary. And there's also increasing interest in employing SIT to combat agricultural pests, like those pesky fruit flies that cause so much damage to crops worldwide.
It sounds like SIT could play a pivotal role in shaping a more sustainable future for pest management. It's about finding ways to control pests without relying solely on those broad-spectrum pesticides that can have unintended consequences for the environment and human health.
I couldn't agree more. SIT aligns perfectly with the goals of sustainable pest management. It's about taking a targeted approach, minimizing our impact on the environment, and safeguarding human health and well-being.
All right, we've covered a lot of ground today. From the fascinating history of SIT and how it works, to the challenges it faces and the incredible potential it holds for the future. I think we've given you a pretty thorough deep dive into this remarkable technique, but before we wrap up, let's do a quick recap of the key takeaways.
Absolutely. It's always helpful to summarize the main points.
First and foremost, SIT is a revolutionary pest control method that uses sterilized insects to suppress or eliminate pest populations.
It all started with the groundbreaking work of Edward Nippling, and the iconic eradication of the screwworm from North America stands as a testament to its power.
And while SIT has achieved significant victories against various pests worldwide, it's not without its challenges, including those posed by the pests themselves, environmental factors, and public perception.
But despite these hurdles, the future of SIT looks incredibly bright, fueled by advancements in genetic technologies and the potential to expand its application to tackle some of the world's most pressing pest problems.
So whether you're a farmer grappling with crop-destroying pests, a public health official battling disease-carrying insects, or simply someone who wants to understand the innovative ways we're managing insect populations, SIT is definitely a topic worth knowing about.
It's a fascinating example of how human ingenuity can be harnessed to solve complex problems and pave the way for a more sustainable future.
Well said. And now I have a question for you, our listener. Imagine a world where we could effectively control some of the most challenging insect pests, using a method that's both targeted and environmentally friendly. With advancements in biotechnology and a growing focus on sustainable pest management, could SIT actually become our go-to solution?
That's a question a lot of researchers are grappling with right now. The potential is definitely there. But we need to remember that SIT isn't a magic wand. It's a powerful tool that needs to be wielded strategically, often in conjunction with other approaches.
So it's about finding the sweet spot the right context for SIT to shine.
Exactly. For some pests, SIT might be the main event, the primary solution. For others, it might play a supporting role, working alongside other methods to create a more comprehensive strategy. The key is to carefully evaluate each situation and tailor the approach accordingly. There's no one-size-fits-all solution in the world of pest control.
We talked earlier about public perception being a potential roadblock. Do you think public engagement will play a crucial role in the future of SIT, especially as we consider expanding its use?
Public engagement is absolutely essential. It can't be an afterthought. It needs to be prioritized from the very beginning of any SIT program. Transparency is key, along with clear communication and genuine community involvement.
So it's about building trust and making sure people understand the science behind SIT and why it's being implemented.
Right. And addressing any concerns about those potential non-target effects is paramount. We need to be upfront about the risks and benefits and involve the community in the decision-making process. It's a collaborative effort.
It sounds like the success of SIT, especially as we venture into new territory, hinges on public understanding and support.
Not a doubt.
Well, I think we've explored just about every nook and cranny of the sterile insect technique today. We've covered its humble beginnings, delved into the intricacies of how it works, confronted the challenges it faces, and gazed into its promising future. I hope you, our listener, have gained a deeper appreciation for this remarkable method of pest control.
From its modest origins to its cutting-edge potential, SIT is a testament to the power of scientific innovation. It reminds us that even the tiniest creatures can play a part in solving some of humanity's biggest challenges.
Well said. It's been a fantastic journey of discovery. Thank you for joining us on this deep dive into the world of SIT. We'll be back soon with another exploration of a fascinating topic. Until then, keep those minds buzzing.